Hello. In my view, translation is, from the evolutionary perspective, the most puzzling or intriguing one aspect in biology. And for this, to understand how the translation as a mechanism has evolved or emerged, and how these ribosomes have evolved, is a kind of very interesting point to understand, because we are talking about a complex that can read the mRNA and then can pick the correct amino acid to come into place. And that mechanism is done by ribosomes, and it's a very fascinating topic to discuss. We are not going to discuss about the evolutionary aspects right now, but we will just see the conceptually what are the ribosomes and how do they manifest what they do. So in ribosomes between prokaryotic and eukaryotes they have lot of similarities, but at the same time a lot of differences as well. As a mechanism and as a structure, they do have similarities in that they are composed of two subunits, and that is, say with the prokaryotes, we have two subunits, and the total of it is about 70 Swartberg units. It's about the sedimentation coefficients, sedimentation rates, and so on. So based on density, and the 70S ribosomes have two subunits. One is 30S subunit, and the other one is 50S subunit, the small and large subunit. Similarly, in eukaryotes too, we have ribosomes with two subunits, but they are a little more complex. They are larger in size too. They're 80S type, and you can see that in 40S, that small subunit is 40S and large subunit is 60S. And the ribosome is composed, it's a ribonucleoprotein. Basically, it's a complex of several proteins and rRNA. That is the ribosomal RNA. Say the ribosomal RNA in prokaryotes here, we have in the large subunit, we have 23S rRNA and 5S rRNA. The small subunit, 16S rRNA is present, and it has several functions, important functions in recognizing the mRNA on the ribosome binding site, etc. In eukaryotes, the small subunit, that is the 40S subunit, has 18S rRNA as opposed to what was 16S rRNA in prokaryotes. And the large subunit also has 28S rRNA, 5S rRNA, and 5.8S rRNA. And in an association, they also have large number of proteins. There are approximately 34 subunits, proteins that are associated with the large subunit, and 21 with the small subunit in prokaryotes. In eukaryotes, we have 49 proteins in the large subunit and approximately 33 proteins in the small subunit. It would be nice to think of the ribosomal proteins in eukaryotes and the organization of the ribosomes. The ribosomes are organized in the nucleolus of a cell. So if we have a cell and we have a nucleus, in the nucleus we have regions that is nucleolus. That is where the genes of rRNA are located or localized, and that is where transcription of the rRNA genes happen, and so the rRNA are produced in that place. And so the proteins of the ribosomes, however, are the mRNA is made, transported into the cytoplasm, the ribosomal proteins are synthesized, they are then taken into the nucleus where the ribosomes are organized. You can have the composition of these proteins and rRNAs separately, and we cannot just dump them and then say the subunits to form. Instead, they should have been sequentially added, and people have worked out how to reconstitute the full ribosomes from their basic constituents. And that's amazing, that how people have been able to make translation happen in the test tube. That is, in vitro translation can also happen. It's amazing to see the formation of ribosomes itself. mRNA of the ribosomal proteins being transported, then translated, and then the proteins carried and ribosomes made, and the ribosomes then transported into the cytoplasm, where they carry out a large number of translation of every other protein as well. I hope we can appreciate the complexity and the beauty of ribosomes. In further lectures too, we will see how well and how amazingly they work. See you later. Bye-bye.